Hello. Welcome to the next in the series of review clips designed to assist you in preparing for your Biology 301 public examination. In this clip, we'll be examining genetic crosses, how to do them, certainly drawing specific attention to the monohybrid cross, or one-trait cross, and the dihybrid cross, or two-trait cross. With all of these crosses, beginning first monohybrid or single-trait cross, you have to initially set up the parental cross, or P1 generation. In this case, the example I'll work through will use axial flowers versus terminal flowers to be the trait in question. If we take a male axial and cross it with a female terminal, the cross would be as follows: big A for axial, cross with little a for terminal. We know then that the gametes produced, which would be step 2, would be big A or big A. Because both of these individuals are pure, they can only produce one type of gamete. Once we have the gametes determined, we now set up the Punnett square, with each of the possible alleles written in the corresponding blocks. For the male, big A, because that's the only gametes that are available, little a. Once the alleles are placed in separate boxes, we can then start to compare them. Big A, little a. Big A, little a, simply bringing the alleles down and to the right. Big A, little a. Big A, little a. All of the individuals, 100%, would indeed be heterozygous. This completes the monohybrid cross for the first parental generation. All of the F1 generation are heterozygous, big A, little a, 100%. In terms of phenotype, the phenotype would all be axial, as axial is dominant. When we cross the F1 generation, the same pattern is applied, except in this case, each individual, because they are heterozygous, has two possible gametes that can be passed on. Each individual can pass on a big A or little a. Big A or little a. Same pattern is followed. Set up your square and you put each allele in the corresponding boxes. You bring your gametes down, big A, big A. Big A, little a. Bringing the gametes to the right or down as appropriate. Big A, little a, little a, little a. When the cross is performed, what we get is one homozygous dominant, one of the 4 homozygous recessive, and 2 of the 4, or 50%, are heterozygous. The genotypic ratio is 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive, for a 1:2:1 genotypic ratio. As for the phenotypic ratio is 3 axial, the homozygous dominant, plus heterozygous, and one terminal, the homozygous recessive, for a genotypic ratio of 3 to 1. In both cases, the phenotypic ratio is 3 to 1 axial terminal, 1 to 2 to 1 for the genotypic ratio. 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. Let's take the following example. We'll do another one through. In this case, brainy bunnies, where big B is dominant. This is the brainy gene. Little b is recessive for not so brainy, for dumb. So we have a dumb bunny and a brainy bunny. The first step, we set up our cross. Because both bunnies are pure, the dumb bunny can only pass on little b's. The brainy bunny can only pass on big B's, giving us the following: 100% heterozygous in our F1. All individuals are heterozygous big B, little b. Convention is that you write the dominant letter first. Dihybrid crosses, or two-trait crosses, are done in the same manner. As before, we set up the parental cross first. In this case, we're going to be dealing with two traits at one time: flying and color. Purple is dominant over orange. Flying is dominant over walking. We're going to start with two pure lines: flying and purple, and orange and walking. When we're doing a dihybrid cross, we'll use the FOIL method to determine the possible gametes. Let me explain this a little further. If our genotype is big F, big P, big P, what are the possible arrangements? We start by pairing the first allele with the two others for the second trait. We do the same with the second allele. This gives us the possibility of big F, big P, big F, big P, big F, big P, or big F, big P. And while in this case, because you are dealing with the pure lines, they are all identical, there are four possibilities. If this individual is heterozygous instead, bear with me 1 second, please. If the individual is heterozygous instead, it would be big F, little f, big P, little p. Using the same method, pairing up all the possibilities, we would get big F, big P, big F, little p, little f, big P, little f, little p as being the possible gametes. So we set up our parental cross. For again, we're writing all the possible gametes from both parents across the top and down one side. We then transfer the gametes through. So this one would be big F, big P, big F, big P, big F, big P, big F, big P, and so on until you are completed. Because both parents are pure, all of the offspring are going to be heterozygous. (100% heterozygous). As you can see illustrated. We then would do the F1 cross. In this case, we are crossing two heterozygous individuals, and we have to choose one of each type of allele, again using the FOIL method as I previously demonstrated. When I do the FOIL method, my possible gametes would be big F, big P, big P, little p. The possible gametes you see listed on the left and the cross on the right. You then would match up the traits, moving through, to give the following. Once you have your Punnett square completed, you then need to interpret the results of that square, writing down the genotype and phenotype of each of the potential offspring. Remember, these are potential offsprings. Punnett squares give you percentage ratios. In this case, we have 9 individuals, which are going to be flying in purple, 3 individuals, which are walking in purple, 3 other individuals, which are flying in orange, and 1 individual which is walking in orange. This 9 to 3 to 1 ratio is characteristic of all dihybrid two-trait crosses between 2 heterozygous individuals. Again, another example. In this case, we are looking at seed color and seed shape. Smooth versus wrinkled, yellow versus green. Completing the square between 2 individuals which are heterozygous yields the following results. 9 smooth and yellow, 3 smooth and green, 3 wrinkled and yellow, 1 wrinkled and green. Again, this is the characteristic ratio you get from all crosses involving heterozygous individuals for 2 traits. Past public examinations have yielded questions on this topic quite frequently. I present the following to you for consideration. In this cross, you are told that black coat color is dominant to white. If a heterozygous black guinea pig is mated with a white pig, what percentage of the offspring should be black? The quick way to do this, the proper way, would be to firstly construct a Punnett square for yourself. You're told heterozygous black. So the male, assuming male, is heterozygous. Big B, little b. White, because white is recessive, In order for an organism to display a recessive trait, it must be homozygous recessive. So the white pig is little b, little b. We start by writing our gametes, possible gametes, along the top for one parent, big B, little b, and on the side for the other, little b, little b. I then match up big B, little b, big B, little b, little b, little b, little b, little b. The question asks what percentage should be black. The following individuals are indeed black colored. Here, and this individual is black. The following individuals are white. 1, 2. 2 of the 4 are black, therefore 50%. The correct answer is C, 50%. Constructive response questions are commonly posed as well. This question is worth 4%, or 2% of your course mark. You are told that in corn plants, thick husk is completely dominant to thin. In all these questions, you'll be told the dominance relationships for all traits. Green seed is completely dominant to yellow seed. That's the first piece of information you need to consider. The second piece of information, and the first tip, what are the parental genotypes? That is the first thing you need to determine. You are told 2 thick husk green seed plants are crossed, and one of the plants is thin and yellow. The key to determine the parental genotypes is this individual, which is thin and yellow. Because both those traits are recessive, one of the offspring holds the genotype little t, little t, little g, little g. In order to have that, it must have gotten a little t from both parents, a little g from both parents. With that information in hand, given that both parents are showing dominant, both parents are big T, little t, big G, little g, cross with big T, big G, little g. 1 mark was awarded for determining this. You are asked to show all workings. The most advisable pattern would be to simply construct a Punnett square. To construct your square, in this case 2 traits, which is what I've done here. Because we are dealing with a heterozygous individual for both traits, using the FOIL method, I know that the possible gametes are big T, big G, big T, little g, little t, big G, little t, little g for both. I would then simply write in the gametes across the top and down the side. Both parents share the same genetic combination. I then fill in the square. Big T, big T, big G, big G. Big T, big T, big G, little g. Big T, little t, big G, big G. Little t, big T, big G, little g. Big T, big T, big G, little g. Big T, big T, little g, little g. Big T, little t, big G, little g. Big T, little t, little g, little g. Big T, little t, big G, big G. Big T, little t, big G, little g. Little t, little t, big G, big G. Little t, little t, big G, little g. Big T, little t, big G, little g. Big T, little t, little g, little g. Little t, big G, little g, and little t, little g, little g. This completes our square. Once you have completed the square, you then need to determine the genotypes and phenotypes of each of the offspring. What you're asked to find in this question is, what is the percentage of the offspring that share the same genotype as the parents? This would be worth 1 percentage point. So you're looking for those that are heterozygous for both traits. So looking at this here, going through the list, we have one individual right here. I'll just mark a 1 to keep myself straight. 1. Here's a second. Here's a third. Here's a fourth. So there are 4 individuals that share the same genotype as parents. You're also asked, what is the phenotypic ratio of the plants produced? There's a quick way to do this. If you recall, you could have counted all the individuals, of course, individually. Or, if you recall, for all crosses involving parents that are heterozygous for both traits, you always get the 9:3:1 ratio, or 9 which are, in this case, thick and yellow, 3 that are thick and green, 3 that are thin and yellow, and 1 which is thin and yellow. And that would be the phenotypic ratio. My strong suggestion for you at the end of each of these questions is on the side, very clearly, write in all your answers. So you'd write in part 1. That way the marker can clearly see it. The question that you were asked originally was, what percentage share the same genotype as the parents? From your square, you determine there's 4 individuals, 4 of 16. So that would be 25% same as parents. You were also asked, what is the phenotypic ratio of the plants produced? What you would do then is, again, the same thing. Part 2, 9 to 3 to 3 to 1, stating 9 thick and yellow, or thick and green rather, 3 thick and yellow, 3 thin and green, and 1 thin and yellow. Make sure that in your examination you write your answers clear so the marker can see them quite readily. If you are asked to show all workings, please ensure that you do so. Thank you for your time. I hope you found this learning clip helpful.